Good morning.
Yes, one second.
Yes.
You got my presentation. ये बाद में कर लेते हैं.
ठीक है. A really great morning to all of you. From the NIT Pharmacy Institute, this is Anmol Dogra, extending greetings to respected Director Abhijit Majumdar, Director, NIT Pharmacy Institute, Dr. Rupa Majumdar, Dean of the NIT Pharmacy Institute, Dr. Ritushri Kupreti Man, prominent guests, and other eminent participants. I am delighted to welcome you all to what's bound to be a remarkable and life-changing ninth session day 5 of the Faculty Development Program, which is sponsored by Dr. APJ Abdul Kalam Technical University. So I am excited to be facilitating this wonderful FDP. Your presence here today demonstrates a common commitment to creativity and the progress of the knowledge. So I want to thank Director Sir and Dean Ma'am in particular for being our sponsors and the partners, as their journals has made our activity possible. Your dedication has truly helped to bring us together, and I appreciate that. So before we begin the session, I would like to invite Dr. Rupa Majumdar, Dean of the NIT Pharmacy Institute, to lead the green welcome to Dr. Rutishri Tukriti Ma'am, Chief Scientist, Genomics and Molecular Medicine Unit, CSIR, Institute of Genomics and Integrative Biology. So I request Ma'am and Ritushri Tukriti Ma'am to please come. A big round of applause for the ma'am. Thank you, ma'am. So prior to inviting Dr. Ritushri Kukreti ma'am for the session, I would like to enlighten ma'am's profile. Ma'am, currently she is Chief Scientist at the CSIR IGIB in the area of the pharmacogenomics. Ma'am obtained her PhD in 1994 from the Department of Biosciences and Biotechnology, University of Roorkee, presently IIT Roorkee. Later, she joined Laboratory d'Oncologie Moleculaire, University of Paris, France as a postdoctoral fellow. In 1998, she joined the Functional Genomics Unit at the Center for the Biochemical Technology, CSIR, Delhi, as a research associate and subsequently joined GenomeMed as a senior scientist. Dr. Ritushri Kukreti is the author of the over 110 scholarly publications and has been granted with the 8 patents in the area of the medical genetics, genomics, pharmacogenetics, and the functional genomics. Ma'am has supervised 20 PhD scholars and has delivered more than 80 invited lectures on the genomics and pharmacogenomics at various Indian universities, colleges, and the international conferences. Ma'am is a fellow of the Association of Physiologists of India for outstanding contribution in the field of physiology and also recognized by the ICMR Dr. Vidya Sagar Award 2012 for the significant contributions made in the field of mental health. Ma'am, also an expert committee member for ICMR task force for the 5 years. In addition, MAM is an active member of the institutional CSIR Integrated Skill Initiative for the Genomics and the Molecular Technologies program for the industrial applications. So, on this Independence Day, Dr. Kupreti has hosted the flag at the CSIR IGIB as a symbol of the freedom. So, likewise, we also want the freedom from the cancer. So now I would like to invite Dr. Kupreti ma'am to please come on the dais to start the session. I request ma'am to come on the dais to start the session.
नहीं नहीं मुझे वही चाहिए
वो वाला भी समझ गए?
एक मिनट बस
Okay, you want the presenter. समझ गए मैम?
एक मिनट आई
हाँ, this is what you want, right?
देखो वरना ही
नहीं, नहीं.
हाँ इट्स बेटर ठीक है थैंक यू it's okay no so do I need mic or is it okay like this mic is important or I'm audible hello that's okay ma'am that's okay Mm. Good morning, everyone. So it's my pleasure to be here and thank you all. Thank you, ma'am. Yes. Thank you, Raki. Thank you, uh, Chandana. So it's like my pleasure to be here and I'm very happy to be with you guys. And I'll be explaining or I'll be uh, telling you my experiences during my journey in IGIB and working in the area of pharmacogenomics. It's a long way. So ma'am asked me for to present a little bit about the cancer genomics and basically the pharmacogenomics and how it is important in personalized medicine. So I'm not a person who is of cancer biology, but when it comes for pharmacogenomics, we do understand that how cancer genomics and pharmacogenomics is valuable for us to understand and to diagnose it properly and giving the treatment properly as a on personalized basis. So when we see this picture, you see different colors are here. So there is red, green, and also the yellow ones. So there are different people with variety of genomics, variety of genome variations here. So how do we understand that? How it is helpful as an application to understand which patient will respond to particular cancer drug or which patient will not respond. So basically in pharmacogenomics, what we understand, there is a substantial drug response differences at biochemical, molecular, and genetic level between individuals during the treatment of diseases. It contributes towards disease severity by developing heterogeneous symptoms and as well as side effects. So you can see here, there are responders not predisposed to toxicity. There are non-responders and they are disposed to side effects. Treatment which is given for responders is the conventional drug with optimal doses, which are always given to the patients, generally with doctor prescriptions. But there are few of the patients wherein the treatment we need to understand which need to be changed as the drug, which will suit the patients, and probably they need to change the doses also. So in terms of drug-specific response prediction based on DNA sequence variation, we term as pharmacogeny. Now, what I will be explaining here, that not always the genetics may explain you. There are certain other things at the end. I'll tell you that how biochemical level or molecular level is important as treatment response understanding. How physiology is important. So a pharmacogenomics is promise of affordable healthcare and personalized medicine. So concept is in a ethnicity matched patient population. Are you able to see this picture? Okay. So here there is a population specific allele or genotype based prediction response at a standard dose, where a mutation in a gene marks the non-response of any drug. So you can see there are a group of patients here. So the non-responders, they are low in number and responders are more in number. So therefore, if you see the global epidemiological data, you will find that most of the population, they respond to particular drug. However, 20 to 30%, sometimes it is 40% too, or 20% or less than 20% population, they don't respond at all. So therefore, those patients are crucial to understand that why they are not responding. So it is important to know that. So basically genotype, phenotype and its distribution, what we understand in epidemiology studies. So here, now next is validation by pharmacogenomic screening. What we expect here. Patient pool based on allele and genotype for drug efficacy and drug adverse drug reactions. Some non-responder patients shows good response when administered with a second line drug. So you give first line drug, then they don't respond. So you give second, another drug there. Alternate therapy, what we call here. Screening these patients based on allele genotype is vital, both clinically and economically. So altered therapy is required or altered dose is required for another set of patients, and regular standard dosing for the patients who are responding. So basically, this is a concept of pharmacogenomics. So across years, we published few of the articles wherein this is not only the genetics, that's what I'll be explaining you through the data, that how The other physiological conditions are also important when you study the personalized medicine. So pharmacogenetics, what we call as influence of genetic variations on drug response limited to few genes. Then pharmacogenomics, influence of many genes or entire genome, their interaction with environmental factors on drug response. So genetics and genomics, it comes for the entire genome. When it comes is genomics. And when you study a single gene or few gene, it becomes genetics. So pharmacokinetics, that means the drug is metabolized. Sometimes the drug is metabolized into active metabolite, or sometimes it is inactive. So itself the drug is important, whether that is going to the target, and sometimes the metabolite which is active is important. So suppose the gene which is metabolizing the drug into the metabolite, which is active metabolite, so the gene is important to study, right? So drug metabolism, that is pharmacokinetics, is important here, and pharmacodynamics, that means drug target interaction is important. So these are the basic things which will help us to establish few of the data set, not all. That is what our experience say. So here, from research to clinical translation, a significant amount of pharmacogenomics research has been reported worldwide, but only a handful of it has been translated into clinical relevance. This gaps needs to be filled. So you can see here how many, you can go back to the literature and see how many are FDA approved drug, how many are the hospitals, they are looking at the commercialized level, how many screenings are being done. So that is very, very less. And how many tests are being utilized for commercial purpose as of now. So they are very, very few. So that means there is a huge lot of gap which needs to be understood at the clinical translation. So probably this is very, very important. And as the pharmacology department, I believe that here the pharmacogenetics, where the pharmacokinetics comes, it is very, very important. How you can set the dose will be dependent on the variations of the genes. So probably this can be a huge success at your department. Only few of the labs across globally, they are looking at pharmacodynamics. So pharmacodynamics labs are very, very few across world. So not much are doing in pharmacodynamics. Most of the work are being carried out at pharmacokinetics level. And it is so huge that there is lot of work needs to be done as yet. So here on screening the global pharmacogenomics literature, biological entities like genes, diseases, drugs were identified in varied populations. These pharmacogenomic relationships are obtained from the identified. You may not see here, but I'll let you know. This is a radial cluster showing global 2304 pharmacogenomics knowledge resource. This we have published in Frontiers in Pharmacology in 2019. And what we showed from the literature, that these 230 pharmacogenomics markers are very, very important across number of diseases. How many diseases? 1753 diseases, 666 drugs, 4132 genes are important, and 33942 polymorphisms are important. From the total publication shows biological entities and their relationships implicated in drug resistance, right? So aim is to develop pharmacogenomics assay panels which can be commercialized. Here it is important to note which can be commercialized for drug response, efficacies, and adverse drug reactions too. We have identified this strategy for identifying potential predictive pharmacogenomic signals here. In this paper, we have shown. You can check this paper and you can actually take up the cancer pharmacogenetics markers from here and you can develop the panels yourself. So, looking at the hospitals based clinicians and industry people who are actually working here. So you can look into these panels and go ahead. Now, pharmacogenomics, biomarkers, and their therapeutic information within FDA approved drug labeling. So you can see here, oncology is the first row there. It has a highly diversified with multiple biomarkers. So there are so many biomarkers, the gene biomarkers, TPMT, CYP2D6, G6PD, CYP2C19, ESR, PGR. So there are many. So you can see oncology is one related area where lot of variations they are present. It's important. So here, next, this is general, which I have just counted here, that cancers refers to abnormal and unregulated growth of tissues which may invade surrounding normal structure as well as spread of to sites distant from that of the origin. Three major stages of cancer hallmarks are in the following order: reprogramming, cell growth, birth and survival, proliferation, invasion, and metastasis. Thus, once the malignant cell is formed and has survived, a first round of cell division, invasion, and metastasis may follow as an inevitable consequences. So if you look upon the types, cancer types, based on the cells, they arise, there are more than 500. There are carcinomas, there are blood cancers, and there are sarcomas. So now here, how many? Nearly 10 million deaths they are reported. The most common 2020 were breast cancer, lung cancer, colon and rectum, prostate, skin, and stomach. In India, most common cancers in 2020 for males, lung, mouth, prostate, tongue, stomach. Now, here a little bit a research thing which I need to tell you here. When it comes that for males, I will take an example of lungs. Why we say for lungs, for males, the lungs have more affected, whatever level, fibrosis, pneumonia, then cancers, they're more common in males. So the research part here is, what are the significant changes between males and females are there when it comes for lung cancer? Why lung cancer is more common in males? It is very, very important for us to understand how mouth is affected more here. Prostate, tongue, stomach. So these are important things which seems you may not understand because these are just the words we have put up here, as in we got the percentages like from epidemiological studies. But when we study here, there's a huge, huge in-depth knowledge is there, which says that why males are affected when it comes for lungs. For females, the breast, cervix, ovary, corpus, uteri, then lungs. Of course, but it is less than males. 1 in 68 males, 1 in 29 females, and 1 in 9 Indians will develop cancer during their lifetime. The projected incidence of patients with cancer in India for the year 2020 among males is 94.1 in 1 lakh, and among females, 103.6 in 1 lakhs. So the most common causes of cancer here, which shows is lung and colon and rectum. Now, cancer genomics, how genomics will help us to understand the development of cancer. So it's a group of genetic diseases that result from changes in the genome of cells in the body, leading them to grow uncontrolled. These changes involve DNA mutations in the gene. Cancer genomics is the study of the totality of DNA sequence and gene expression differences between tumor cells and normal host cells, the germline, and somatic mutation. It aims to understand genetic basis of tumor cell proliferation and the evolution of cancer genome under mutation and selection by the body environment, the immune system, and therapeutic interventions. Cancer genomics focus on finding new cancer genes, identifying molecular signature to stratify tumors, which is very, very important for personalized medicine. Stratify means you need to segregate them based on the mutations, finding new therapeutic targets, and move towards the personalized cancer treatment. Now, from 2304 pharmacogenomics markers, 336 markers are identified for FDA approved drugs, which are 207, showing ease in clinical decision, making the value addition in healthcare. Here, if you see cisplatin, the drug which is given for cancer, the TPMT gene is very, very important, wherein TPMT star allele 1, TPMT star allele 3A, 2A, which are very, very important, which I'll take forward next. So here, if you see, out of 2304 pharmacogenomics markers, only 15% of the markers, they are being commercialized in hospitals where they do diagnostic tests, or industry, they use the panels and all, but still 85% is left. So we need to work for all these markers to check that how these markers will help us to understand the diagnosis of the cancers or the efficacies, the drug responders. So it's important. So whether these markers which are present in this rows, how they will help us for understanding whether patient is responding or not responding. So this work is left, and we need to make the utilization of all the markers which are present globally, because this is a global data which we have collated in a very systematic way. And this is what I was telling, that there is so much of data which we need to understand and put together where there is no fund is required. So we can make those kinds of databases, and then we validate that single and utilize, commercialize. So you can get the money also to further do the validation steps. Now, this slide will tell us that some of the FDA, Food and Drug Administration USA, approved drugs with their clinically tested markers for testing drug response, better efficacy, and least toxicity. So here there are a lot of drugs: carbamazepine, warfarin, clozapine, abacavir, clopidogrel, azathioprine, irinotecan, and 5-fluorouracil. So I'll talk about azathioprine is an immunosuppressive antimetabolite drug used to treat autoimmune condition. It is known as antimetabolite due to its ability to form toxic purine analogs. Just one. So antimetabolites are chemotherapy drugs that prevent cancer cells from making more cancer cells. Lymphocytic lymphocytes replicating also. They trick cancer cells into using the drug instead of the molecules it needs to make the genetic material to replicate. And this is the way it suppresses. So here, if you see the recommended commercialized markers which are being used for TPMT genes. So I'll take up this thing. The azathioprine drug is upon metabolism is converted into 6-mercaptopurine, which is further converted to a toxic purine analog 6-thioguanine that inhibits DNA, RNA, and protein synthesis. Now, here, inactivation is done by TPMT gene. So this is important. So there are 3 alleles here, which are star 2, star 3A, star 3B, and star 3C, which are very, very important. Allelic variants with reduced or no activity of TPMT gene. Variability, this shows the variability in metabolism, and therefore the variability in response will occur. So if you want, I can take up more understanding into it. So when there is a wild type, it is extensive metabolizing for the drug azathioprine, which is making the 6-mercaptopurine. And then with mutation, it is good responders. And if both are there, it is poor metabolism and there is a drug toxicity there. So here you can see the dose requirement. When there is extensive metabolizer, the high dose is required, then intermediate, and then low dose is required. So you need to adjust the dose according to the TPMT variation here. So you can see here. So this particular 6-mercaptopurine is actually inactive metabolite. And therefore, it is important through TPMT to balance the effect of azathioprine. So these 3 account for 95% of clinically significant mutations resulting in low or intermediate enzyme activity. TPMT polymorphism and risk of toxicity. As mutant alleles increases, TPMT activity decreases. You can understand that. If there is wild type, there is a high metabolism. And if it is mutant allele there, it is the TPMT activity decreases. And the TGN accumulation increases, resulting in myelotoxicity. When the full dose of azathioprine is given to a homozygous mutant, it can lead to toxicity as 6-mercaptopurine will be accumulated in very high amount. For homozygous mutation, TPMT activity is very low and TGN accumulation is very high. Now, this is the work what we have done in our lab, the cisplatin ototoxicity here in pediatric patients. So what is here? The distribution of TPMT 3A risk genotype, 3A. Here we are showing only 3A risk genotype in Indian population. So we screen the entire Indian population, and what we see here, the mutant allele is present here only in one of the population, which is Northeast population, and no one else. So basically, the entire Indian population is prone for high metabolism rate for azathioprine. So we need to be very careful about the doses given to the patients suffering for the cancer here. Showing measures of clinical utility and potential population impact. So wherever you want to plan for the panels, you need to understand which population you are doing and then make the panels there with mutation allele, wild type and intermediate. Then you will automatically will see that the population based genotypes, they will tell you the risk content here. So the major complications that limit the use of cisplatin is the risk of drug-induced ototoxicity, which manifests as permanent bilateral hearing loss. So therefore, it will help further for the ototoxicity. So cancer-associated mutations are oncogenes, tumor suppressor genes, DNA repair genes. These are very valuable, and you will find that the mutations carrying these genes will help you to make the panels, which will be taken up by the population studies. Carcinogen, activating genes, deactivating genes, cell cycle checkpoints, cell death genes, cell signaling genes, differentiation, senescence genes, metastasis. So now currently the genetic testing for breast cancer, what are the panels which are people are selling? High risk genes are BRCA1, BRCA2, PALB2, P10, TP53. These are being commercialized now. Moderate risk genes for breast cancer, low risk gene for breast cancers. So these are the panels which are already there in the market. Genes with insufficient evidence for breast cancer risk. Then there are a number of other genes, including the CYP19A1, FGR, R2, H19, TERT, that are thought to also increase the risk of developing breast cancer when they carry a mutation. This is just a glimpse for you people to know the major events in a decade of cancer genomics. So here the major advances in massively parallel sequencing platforms and targeted enriched technologies, major large-scale project designed to catalog genetic or genomic markers of normal human individuals. So this is important. When you say there are normal individuals, we want to tie the genomic variations. So how much is the genome? So it's a huge genome. How many base pairs are there? 3 billion base pairs. And when we come to understand the variations among the human normal individual, it comes 30 millions of variations as of now is being reported. Now, what is there? There are so many millions of variations across populations. So we need to understand, we need to make the databases according to the pathways, according to the way I told you earlier, that we need to have the set of genes where there are cancer genes, lot of different variety. We can tabulate our database accordingly. So, but how we will do it? We need to have the technologies for understanding genome variation. The sequencing is the very, very important technology. So cancer genomic databases of single nucleotide polymorphism, haplotype map of human genome is being tabulated as of now. Then there is one more, very important, the ENCODE encyclopedia of DNA elements, which is actually helping for activating the genes or deactivating the genes or productions of lot of proteins which are involved in the cancers. So that is what ENCODE and encyclopedia, then cosmic catalog of somatic mutation in cancers. So it's very important. Somatic mutation, identifying it is very, very important, and it is difficult too because each individual will carry different somatic mutations there. And then cancer genome atlas, genome analyzer, colorectal carcinomas, whole exome sequencing. So what is the difference between next generation sequencing and exome sequencing? I hope you may be knowing it, that the next generation sequencing or the sequencing technology covers the entire genome. But when we say whole exome sequencing, only the exomes, they are being sequenced where they codes for the proteins. Then International Cancer Genome Consortia is being developed, tumor sequencing projects, acute myeloid leukemias, whole genome sequencing, ovarian small cell carcinoma being identified. So these are the major events in the decode of cancer genomics. It's very, very important, and everybody should go through all these papers, which are the major milestones in cancer genome. Without these papers, you may not be able to understand that how it is being developed, the roadmap of cancer genomics. Then the timeline for most of the remarkable precision oncology highlights the Herceptin trastuzumab map, 1998. So you must be aware of Herceptin here. Then ER, tamoxifen. So these are some of the major the drugs, the genes which are being identified. So study evolution in precision oncology in the last few decades, a landmark therapeutic molecular targets for common anti-cancer drugs, identified the different diagnostic technologies, then regulatory landmarks. Regulatory policies are very, very important here. We ourselves cannot commercialize the genome variations panels. So this is very, very major. Do our Indian government have that clue that we need to understand if we are commercializing, how come Gangaram is doing lot of significant genome variation testing. So it's important. So there are certain regulatory aspects wherein our clinicians play a major role, which are the authorities to say how important it is to check the genome variations. So we need to understand the regulatory policies of our Indian setup to commercialize these markers. So therapeutic landmarks and their molecular targets, most relevant diagnostic technologies and regulatory landmarks are important. Then Foundation 1 is the first FDA approved qualitative NGA-based in vitro diagnostic test for advanced cancer patients with solid tumors. The next is the impact, MSK. So Memorial Sloan Kettering integrated mutation profiling of actionable cancer targets. So it's important to know all these things, which will really help you to go with the landmarks, milestones. And now next what, where you need to put your efforts at in Indian scenario to go ahead is important. So cancer projects and consortia which are present here, which will tell us that expression patterns, this project is the expression patterns. What are the expression patterns of the genes? Copy number alterations, somatic mutation, epigenomic profiles, structure aberration, germline way. So in systematic way, the entire energy, the entire values given to different level to sort out, to understand cancers is important. That's the reason such projects and consortia are built. But not everything is being done by the same person. Different people in consortia do the work with the same patients. So they club the data and then they come out with the significant discoveries there. So basically it shows the cancer harbor germline variants here. And it's a Nature communication paper in 2020, which is showing the cancer projects and consortia. Here, this is Cancer Genome Atlas program. It's a major program which is known as TCGA Pan Cancer Project. Here you can see mutation, the same thing which the previous slide was telling, mutation, copy number, gene expression, DNA methylation, microRNA, then clinical data. So the TCGA Research Network has profiled and analyzed large number of human tumors to discover molecular aberration at DNA level, RNA level, protein level, epigenetic levels. Data provided a major opportunity to develop an integrated picture of commonalities, differences, and emergent theme across tumor lineages, that how the tumor is being developed here. Analysis of the molecular aberration, their functional role across tumor types is helpful to extend therapies effective in one cancer to other with similar genomic profile. It seems to be very simple, but again, I will tell it's not that easy. It's really tough here. So to have this understanding, you can see when it comes for personalized therapy, the cancer causing mutation with drug treatment available according to the mutation is important, but we don't have much. We only have, for example, BRCA mutations and then Herceptin, PIK3CA mutations. So not much is being done. There's a lot of things where if we work, we will get roadmap and guidance how to treat the individual patient. Mutations are there with no drug available. Then cancer drugs are available for few of the cancers only. So radiations and chemotherapy is important in cancer. So I'll just go through a study which I've not done in cancers, but it is important to show you here that how personalized medicine is important in case of any kind of disease, any kind of disease. So this is a data of differential gene expression profiles. The analysis is being done in blood, and there are three types of patients. There are idiopathic patients, there are cryptogenic patients, and there are symptomatic patients. So the gene expressions was come out that there are idiopathic patients having epilepsy, were compared with when healthy individuals, we found the number of patients, a number of genes were diversified. There are a lot of, where genetics probably is involved, but we are not able to understand what genes or what will define the idiopathic. Then there are symptomatic patients, the cryptogenic patients. So when we go here, so you can see here differential expression patterns are observed with the highest number of genes in the idiopathic epilepsy patients. Now next is when we give the drug to the patients, the gene expression analysis for valproic non-responder patients versus drug-free patients and valproic responders versus drug-free. So these were compared And what we observed here, you can see that valproic responders versus non-versus with drug-free, you can see the yellow color is drug-free patients. Then valproic non-responders are having some orange color, and the blue color is valproic responders. We show that gene expression studies are important, wherein we understand the different gene expressions with responders, poor responders, and the people who were not given the drug. So if you see differential gene expression profiles in non-responders, you can see here there are certain genes which are tagged here. Element genes granulysin is the gene which we have quoted at the top. We showed the p-values is very significant. Then FDR value is significant. Fold change is very high with differences in the non-responders and the other part responders. When we check the differential gene expression profile in responders, now you see it's interesting here that the first top gene, which were highly significant as p-value and fold change, what we observed here in responders, there is a fold change decrease in responders of prostaglandin synthase 2. So this is very, very important here to understand. So what we did, this gene is PTGS2, seemed interesting to study as it synthesizes prostaglandins, which are easily measured in plasma. So now what we did next here, plasma prostaglandin PGE2 profiles in healthy controls and patient groups were measured. So you see here, just check. The plasma was checked for valproate responders. Lower PGE2 levels were observed in the responder case. Directly we did plasma of patients. So you can see here that the PTGX2 here, it is found to be lower. Then what we did further here, the PTGX2 encodes, this is a literature we observed, PTGX2 encodes to COX2 enzyme, catalyzes the conversion of arachidonic acid into prostaglandins. So you can see arachidonic acid, COX2, it changed to PGE2. Now, when we give, so this PGE2 binds to EP1 and then ABCB1 is activated. When ABCB1 is activated, it is down-regulating. When we give the valproic, it is down-regulating PGE2, glycoprotein. So what the meaning is that PTGX2 encodes to COX2 enzyme, which catalyzes conversion of arachidonic acid into prostaglandins. But when valproic is given, this prostaglandins is lowering down, and therefore, based on that, when there is low of prostaglandin, it will not affect the PG glycoprotein. Now, this PG, which is present in blood-brain barrier, the efflux of the drug is more outside and it is not reaching to the, when it is more. And if valproic is given, the PG2 is less, and therefore what happens? The drug is reaching to the targets. It is not coming out. So therefore, the valproic patients, they are responding. So basically, what is there when the patients are having high inflammation, the more P-glycoprotein is present there, and therefore the drug which is there, it is not reaching to the target. But if you give not the other drug, carbamazepine, there are a lot of drugs, but if you give those drugs, it is not, still it is refractory. But when especially valproic is given here, the P-glycoprotein, because less of PG2 is happening there, and therefore low P-glycoprotein is present, and therefore valproic is reaching towards the target, and the patients are responding. So basically, it is not always the genetics, it is the physiology also, and drug is also important here, that which drug will respond by the patients and what is there, the physiology. So valproic inhibits COX-2 activity, affects the drug efficacy by regulating multi-drug resistant transporters. Just skip this. So the clinical evidences are here that NSAIDs as adjuvants in patient experiencing seizures. So whenever you give the drugs to patients who are having seizures, the patients do not respond, but they are having seizure. But when you give NSAIDs, which are anti-inflammatory drugs, along with the main drug, they start responding. So this is the literature, collated literature, which shows us that patient studies show potential clinical use of NSAIDs, non-selective COX-2 inhibitors, as adjuvant as a future strategy for epilepsy treatment. So it is not for this particular disease or only. You can give for n number of diseases wherein inflammation is high and the drugs, the response to that inflammation is so enormous that there are certain pathways which are activated, and therefore drug is not reaching to the target. So you need to give anti-inflammatory drugs with the main drug so that patients are going through less of the inflammation and therefore they start responding. So personalized treatment, not always genetics, but there are a lot of biochemical, molecular level and physiological level. We need to understand the patient as a whole. Clinical evidences of probiotics. Now, how probiotics are important for all kinds of diseases. This is also given as adjuvants patients. This is especially I have made the evidences collected for the particular disease, but you can see the probiotic administration increases. This is very, very important work, and probably this can be done for the cancer patients, that if you're serving cancer patients for probiotic treatment and including the main drug, you may find better results for that. Why? Because probiotic administration increases antioxidant status and decrease in pro-inflammatory cytokines there through greater production of metabolite like fatty acids, serotonin precursors, GABA, etc., thereby exhibiting protective effects against the disease symptoms. So the approach for disease type, drug response, the marker development, you can see here clinical. You need to understand demographic factors, environmental factors, disease phenotypes, serum drug levels, transcriptomic factors. You can see gene expression profile, RNA sequence variation, genetic, the exome, whole genome profiles, SNPs, CNVs, mutations, and therefore, as a correlation, you understand the drug response. So genomic and pharmacogenomic signatures may elucidate the effects of genes' genetic polymorphism upon individual variations in disease drug response to improve the types and pharmacovigilance capacities of drug responses. Getting to the point, where you could actually use the genetic information to make a prescribing decision requires an extremely high level of evidence, replication, validation, and mechanistic studies. So we are also performing mechanistic studies like drug resistance, studies which I've shown here. Throughout the work, we have done a lot of resources, publications, patents, and detailed phenotype patients were collected. Creation of catalog of prioritized genetic variations is developed. Lot of databases are developed. Generated genomic data, global meta-analysis of drug response studies being done. Establishment with collaborations for multi-centric perspective studies are being done. Functional assays, studies to assess the impact of biomarkers being done, and the chip development is being followed up here. So there is a summary of identifying genetic markers of drug response in Indian populations, similar phenotypic definition across cohorts, provision of replication in large populations, develop therapeutic dosing algorithm, which is very, very important. So this should be the major work you can come out in your centers, where the different dosing algorithm you can develop and can be taken up, where nobody can actually contribute as much as possible from this place, which can be based on age, gender, seizure type, genetic background. Reduced rate of adverse reactions can be performed based on this. So these are the some treatment goals. So these are all some of the publications and knowledge. So all our clinicians, academics, industry, our foreign collaboration, international collaboration, funding agencies, our director. Thank you. Sure, sure. So as far as I understand your question, that sometimes a person which is having a cancer, it may not coming up in the next generation, but may come in the further next generation. It's possible. It's possible because the lot of genetics we are not sure. We only understand the Mendelian inheritance patterns, which shows that next generation to next generation, how the schemes of the elements are passing, right? So there are certain rules which are being mentioned, but there are certain genetics where we are not aware that how that element is over-represented for our next generation. So in that case, you need to be careful, but people generally, they are not very keen to go for the diagnostic test and all. So basically, during that course, I prefer that it is important to go for panels checking, the expression study, and be careful that whether you are having some kind of risk can be present. How much time does it actually take to develop a personalized treatment, for a particular person or patient? See, we seriously look upon very focused project, very focused. So I believe, see, there are two things that we, first of all, we develop a project wherein we see that we need to collect Indian population data. So it's a huge 10 years program. It's a huge 10 years program that we will go collect ourselves. We need lots of resources. We need lots of facility to have that kind of data in place. And working with patients, working with clinicians, it is not easy. They follow up. So it's a huge 10 years, 15 years left.
If the patient is in advanced stage,
Yeah.
then
Yes.
that development of that personalized to customers.
Yes. So I'm telling you the next step, that suppose we are not be doing this, but still we want to provide a better at our end, a better panel, so that they can at least check the risk if it is present there, so that it may help us for particular treatment. So that can be done at, yeah.
It will be advantageous if it is means that part is given at the early stage of cancer. If we start from the early stage of
Yes, yes. So that probably the panel will be utilized for all individuals before also.
similar
Stimulation. So for that case, we need to have a hospital based where the tests are being regularly done. So we have to collaborate with them. We need to collaborate with the industry too.
Yeah.
Those will take us our panel. They should be ready to take up our panels. Then, because we will give to the hospital, clinician has to recommend that, and industry should sponsor that, and then we need to give our knowledge to them. So we will also,
separate research.
but that you will not collect the patients and do your own. You can do this work based on the global evidence. Yes. Where, yes. So those panels, like the work which I have shown here, 2304 pharmacogenomics markers, we can have more stringent analysis also for only for specific cancers, and then make a panel for that and take it.
Worry,
So very,
worry. Because,
very,
yes, because there are three things I'm sharing. First of all, earlier time, there was a very simple classification of cancer stages,
very,
and now they have changed the classification. In next 10 to 24, 20 or 30 years, there will be a new classification. And 20 years back, you know, only there are about 150 or 200 types of cancer. Now the number is actually increasing. So if we, what I am worried, because next 50 years, there will be a lot of new cancers. Now I'm
you
like,
know why this is happening.
it's a very good.
No, you know why this is happening. There are lots of somatic mutations. Germline mutations, you understand that. It passes from generation to generation. They are in the somatic mutations, they are particularly the only mutations. That particular area of cancer. So it is little, it is difficult to understand the heterogeneity there with the patients. So my suggestion for this query is, first of all, you need not to worry of solving each and every confusion at all. Take one release test and understand things and try to solve. Because it will remain on increasing and increasing. But at your level, it is important to contribute. Of your contribution, you cannot solve each and every problem. Right? So, in not to worry, because there's a huge, enormous work need to be done. Not really cross work. So, at your level, your circumstances, your premises, your facility, based on that, you do brainstorming sessions. All people have brainstorming session that what is good at your level, how you contribute and show the industry that this is what we have generated for standard and we are giving. Commercialize that. Immediately you will get enthusiastic about how to go further. So first start with small field, make it successful, and then go.
Well, let's move to our zero function and loss of function studies that we
Yes.
are doing. So then, is it possible by doing such studies to predict that not only the new generation cancer, if you can predict that it will be able to work on this in terms of this.
It can be one.
Particularly for
Yes.
because for the
Yes.
we can evaluate
Yes. But it may not be true every time. That's what is genetic. You may have a kind of mutation there, but you may not be suffering from cancer. This also possible. It is not always like this that you have mutation, which is this mutation, and you are having cancer. ऐसा जरूरी भी नहीं. Because such cases, when you come across, now you understand that how different scenario is. Those mutations like sickle cell anemia, where clear-cut mutation is there and clear-cut phenotype is there. Those are very, very important things, but in cancer, because it is so heterogeneous, you may not find that clear phenotype is phenotype. So we
To give suggestions because
suggestion is this, we need to work with the risk elements which I am mentioning here, which will help us to understand and run for drug responses, but we can also do for diagnosis of disease, and then you can find out those as risk element, find the correct diagnosis, and then run. So you can pick up those variations which are globally defined, that this is the important risk which will cause the mutation, it will cause the cancer. You pick up those first. Go with them.
हमारे देश में तो स्टेज 4 पे बहुत जाते हैं तब पेशेंट्स काफी बदल मेरे दिमाग में चल रहा है इज पॉसिबल कि जब छोटा बच्चा है व्हेन वेरियस गेटिंग बॉर्न दैट टाइम इट विल मेक अ फैसिलिटी डॉक्टर विल टेक द सैंपल ऑफ द पेशेंट एंड आफ्टर इज इट पॉसिबल
ये ड्रीम क्योंकि जो चेक है ना ऐसा बोलते हैं डायरेक्टर wrong that. So our director wrong that was having a dream that everybody will carry a chip of their own name.
चलो
So it will definitely people are working for a like jobs and where you should working on if you want to have that kind of approach. And you need to be clear with that approach and how you go there. And you can do one thing. You need to work. You need to have.
also will solve not only the problem of cancer, it will solve the problem of other diseases.
Let's take examples. Lung cancer. So lung cancer. In lung cancer, the lung fibrosis is there, which is irreversible to treat. Once the fibrosis is done, you cannot reverse it. Ultimately, the patient will die in 15 minutes. To understand in COVID, what happened? Pneumonia and then fibrosis happened and then patient died. Lots of people got pneumonia in the hospital and eventually fibrosis and they died. It is irreversible. Correct. So people are waiting for the drugs which can reverse the disease. I'm working in one of the projects called lung cancer.
we are also working on drug discovery. Very fun. We completed drug discovery. We working on structure based 3-month target databases, screen methodology, so but problem is based on so many years with that. After certain years, after some years, this receptor getting mutated and drug resistance will get developed. So, so then right now, what we see,
How do you expect that mutation to happen? It will take years. It will take why it will happen if suppose there is a mutation? So it will work like before, definitely. But any mutations can still work something wrong. So that is it.
right now we are learning about CAR T-cell therapy and NPT drug. So what is what they are believing? They are believing that CAR T-cell therapy and NPT drug was used better than small molecule based therapy. So are we right or what is the small molecule one in either way?
Yes.
Is it possible if we send you the molecule to a small lung cancer? We present them molecule hospital that be
I will ask. They are ready to with the boundary.
because I'm doing the study, but maybe it's this is can provide the
But there is lot of background work, what needs to be done. Don't start screening.
because we have right now one center taking all of those various targets as well the lung cancer. They have selected one that.
5 kilometers. Definitely. 8. So I mean, there. So I find
Was very wonderful. You did masters right now was clear about that.
Duplicates by the मैम ने डॉक्यूमेंट्स की क्वालिटी पर कहते ग्रुप में डाल दीजिए अब ग्रुप में कुछ नहीं चलिए बोल ग्रुप में डाल दीजिए So thank you, Dr. Preti ma'am, for providing the deep insights of the concept of the affordable healthcare and the personalized medicine, the wide journey from the research to clinical studies, cancer pharmacogenomics markers and the different mutations associated with the studies, cancer studies obviously, and the synergistic effect of the probiotics with the main drug for the better effect. So your contributions, ma'am, have not only strengthened our talks, but have also set the road for the fresh insights and the future developments in the cancer genomics and the pharmacogenomics towards the personalized cancer medicine. So I would want to express my sincere congratulations and warmest gratitude to Dr. Kupreti ma'am for extraordinary efforts that have been critical in establishing the agenda of this FDP and guaranteeing its success. Now, I invite Dr. Rupa Majumdar, ma'am, Dean of the NIT Pharmacy Institute, to present Dr. Kupreti ma'am with a souvenir as a show of gratitude and thanks. So a big round of applause for the ma'am. Thank you, ma'am. So I request to all the delegates to fill the feedback form that is already included in your kits and submit to the registration committee after the last session. So the lunch will be ready by the 1:30. So all you can proceed by 1:30. Thank you. I request to all the delegates to come at the forward to have a group photo with the ma'am. Please come forward. Side करते हैं.
मैम तो फोटो आ रहा है They all are very good and satisfied. I'm very positive. Very positive. ऐसे उल्टा भारत से दे सकते हो ना उससे नहीं बचा
Excuse me. I request you all to please gather at the ground near the stage for a group photo again. मैं वापस क्यों रही हूँ ये बताओ पहले. जी मैम. सर ये है. सर शेयरिंग स्टॉप कर दूं. हाँ शेयरिंग स्टॉप कर दो और सर दीजिए. आप क्या बोलते सोचो कल क्या बोल रहे थे. आपने कब देखा है सोते हुए. ऐसे ही सो जाओ ऐसे सो जाओ. है क्या.